Now this is a pure genomics data set. It's a single cell RNA sequencing study of about maybe 1,500 cells from a brain tumor, and they were just dissolved in solution and flowed through a single cell sequencing kit. So they don't have any actual anatomical coordinates, but the 3D positions of the cells are derived from a UMAP embedding of the high dimensional connectivity structure of the co-expression network analysis into a 3D space. So that's a tSNE right there, and I can also view it as a UMAP. And on the left, I'm going to have the UMAP embedding of the cells. On the right, I'll have a UMAP embedding of the genes. Actually, it looks like I lost a bunch of genes. I'm going to do a tSNE embedding of the genes. There's a lot more genes there. And I'll also do a tSNE embedding of the cells on the left. And right now, the color coding is fusion annotation for the cells. It's we have no fusions with that pink color, and the purplish color is a cell that's harboring an NSF-LRC3783 fusion. So I can blur out the cells that don't harbor any fusions, and you can see that there are a number of cells that harbor a gene fusion there between these two genes, NSF. And then the other colors are the WGCNA analysis modules for the genes, which are represented as these cubes. So we can click on some of these cells and take out the skybox. It's a little distracting. We've adjusted the sizes of the glyphs, so we can click on some of these fusion harboring cells and find which other cells they share a lot of gene co-expression profiles with. And you can also see edges going from the cells to the genes, where the genes represented as cubes shows you exactly which genes are expressed by which cell. So now that we have a bit of a graph edge connectivity web enabled, we can maybe change from fusion coloring to a WGCNA module coloring and see what what sorts of modules are expressed by the fusion harboring cells. And turn down the opacity and crank up the animation so you can see which ones they are pretty easily. Looks like it's this kind of greenish module has the most connections. So now you can switch threshold mode and turn off the intergroup edges. We can maybe click on some of these green genes and light up their intragroup connection network. And maybe make some of these a little smaller. A little smaller. You really see the connection network there. There's a lot we can do. We have connectivity range coloring, so we can limit our searches or our visualizations, or maybe gray out some of the cells with a low number of connections, so that we can kind of get an idea of what hub genes might be in. Well, these are hub cells. These are the hub genes. Those would be the hub genes that have a lot of interactions with a lot of other genes. So yeah, the sky's the limit. Thanks for watching.